All right. Well, I'm going to say, go ahead and get started here. First, I'm going to say good morning, good afternoon, good evening, depending on where you are in the world. And I want to thank you for joining us for the latest installment in the webinar series of the International Pest Risk Research Group. I think we're on our 15th or 16th webinar in this series. And so we're really glad to have this opportunity and get a chance for people to see presentations from the pest risk analysis and pest risk research world. So we're so glad we get volunteers who are willing to do this for us. Come on and do these webinars. And my name is Frank Koch. I am the current chair of the International Pest Risk Research Group, or IPRRG as we call ourselves. And today we have with us 3 presenters. I'll just go ahead and list them off. Let's see here. Give me just a second. Okay, so I have a title in front of me now. Very good. All right, our presenters today are Andrea Maiorano, Eugenio Rossi, and Alex Gobbi from the European Food Safety Authority. And they'll be presenting on climate suitability analyses at EFSA, core activities and future challenges. And I'm going to go ahead and turn it over to you guys so you can introduce yourselves. You can do it in the middle of you can do it now, or you can do it in sequence, whatever you want to do. But the floor is yours. And again, thank you.
Hello, thank you very much, Frank. And first of all, thank you for inviting us to give us the possibility to present our activities, to give you an update on what we do and what we have done above all during the last year, and then presenting you, also giving you some hints about what are our, let's say, daily work challenges and future activities. So I am Andrea Maiorano, and I am the coordinator of this little team, or the coordinator of the activities related to climate suitability analysis in the context of pest risk assessment at EFSA. And so you can see in this room we are 3. We are a small team, let's say, but I mean the climate suitability team is not only us. We have also external collaborator that support our work and support our activity above all in literature search and data extraction. But this is something that you will see during the presentation. So I am Andrea Maiorano. I'm in the room with Eugenio Rossi.
Hi. Hi, everyone.
And Alex Gobbi.
Hello, everyone.
And so I leave the floor to Alex to present the first part of the presentation, which will be followed by, let's say, practical example by a case study by Eugenio. Just a question, can you see our screen? Can you see the presentation?
I can see your screen, but not the presentation.
Yeah, agreed.
Hold on. Can you see the presentation now?
Yes, you just need to put in slideshow.
Yeah, thank you.
Perfect.
Perfect. So thank you, Frank. Thank you, Andrea, for the introduction. And thanks for the invitation to this webinar. Today we will present the climate suitability at EFSA, the activities that we do. So the presentation will proceed as follows. The beginning will just give you a little bit of context about the climate suitability at EFSA, and then we will give you a general workflow that we do, distinguishing between the 2 main activities that we perform, the pest categorizations from one side, and then I will give the floor to Eugenio for the quantitative pest risk assessment, which will be followed by a case study in which you can see some more details of the climate suitability and then some output statistics and future development that we plan to introduce in our group, in our activities. So just to give you a little bit of context, in our unit, we do a lot of pest risk assessments. And among the different aspects that we study, the risk of establishment is one of them. And this also depends, among other things, from the climate suitability, which is the main part of our activity. To do climate suitability analysis, we follow like some sort of workflow or a pipeline that I will shortly present, that starts from a systematic literature search before moving to like a data extraction of different aspects that we are going to investigate later. And then we will produce the first climate suitability product, which is the Copenhagen climate match and climate classification. So the pest categorization, one of the activities that we do, stops here, while for quantitative pest risk assessment, we move forward and we apply different climate suitability models, depending on the needs that the working group asks for. Finally, all the results will get published and shared either through the opinion of EFSA or like some research data repositories that I will show you later. The first step is the literature search. So this is like where we first meet our pests. And to do that, to do like a proper literature search, we need to first collect all the possible names that the pests appear on in the literature. It can be either like different scientific names, all names, synonyms, or like for some plant pathogens, also like common names are very frequent. So we move across like different websites like the EPO Global Database, CABI, and we collect all the possible names until we build a string like the one you see on the screen. Then we run these strings on different platforms such as Web of Science or Scopus, and we retrieve all the papers we can find. We process these papers under two-level screenings on an external platform called Distiller. The first is like based on title and abstract, where we mainly decide whether or not the papers are relevant for the pest categorization or the QPRA that we are doing. And then we move into more detail with the full text analysis, where we extract all the information that we need. During this literature search, we perform a very important step, which is the data extraction. The data extraction is different depending whether we are doing a pest categorization or a quantitative pest risk assessment. The main difference is that our output count for around 40 pest categorization per year against like a 4-6 QPRA per year. And the amount of job is very different. Therefore, like we need to decide what to extract and for the different purposes. The main information we extract is distribution, which we then use for the climate suitability. And we also collect the host range for both the QPRA and the pest categorizations. For QPRA, then we move to some more detail with the pest biology, like the life stage of the pest and which part of the plant was colonized by it. Information about the impact and the spread, and then also about the control methods that sometimes gets applied in order to control the pest development. The first product that we produce into the climate suitability analysis is the Copenhagen, which some of you may be familiar with. We basically put together all the distribution data that we collected, and then we overlap them with the raster of Copenhagen and filtering out all the climates that does appear in Europe. So we only retain the climates that appear in Europe if the distribution if the pests appear to be present in those climates. This is the like at this point, of the Copenhagen point, we stop for the pest categorization. But when it comes to QPRA, we also apply other climatic analysis models in order to deepen our knowledge and about the pest. And so this is an overview of what we already used in some of the recent QPRA that we've been doing. So we applied some SDM, species distribution model, Margaret's model, Climax, and also some physiologically based population dynamic models. Other climatic products that we do only for actually pest risk assessment are like degree days, frost days, isotherm maps, and other customized analysis when required. At the very end, we share and store the research data on a different platform, which is Zenodo. Like here, we put like everything that is not already appearing in the opinion from EFSA. And a good thing of this one is that it allows all different formats and it allows versioning, which means that we can actually update these informations if something is needed. And yeah, so these publications is not intended like to replace the EFSA publication, of course, but it's just like an integration to it. It acts as a complementary analysis where we can also share like all the activities that we do to increase transparencies. Moving into more details to one of the main activities that we do, the pest categorization. So this one proceeds as follows. As we have seen before, we have a literature search, title abstract search screening, the full text data extractions. Then we perform observation quality checks, mostly on distribution, and we build the Copenhagen maps and the deliverables. About deliverables, let me just show you how they look like. So we have first deliverable, which is called the Prisma diagram, which gives information about the literature search, such as how many papers we extracted, how many papers we analyzed, how many papers contains information about distribution, about host range, and also like about some physiological parameter of the pest. Then the whole distribution data are collected into a file called an observation file. Here we only collect information about where the pest was detected, and we consider a detection only when it comes to natural occurrences of the pest in a field, for instance. Another file that we obtain at the very end of this analysis is the master file, which we extract directly from the platform called Distiller and contains information about all the papers that we analyzed, like author, journals, and titles, and all these bibliographic informations, but also contains the host range. So this is also very valuable information to give to the working group. And finally, as we mentioned before, the last deliverable is the Copenhagen map, so which is the first product that we produce in climate suitability. There are like 3 different ways in which we build Copenhagen maps for the distribution data. So these are presented here in a decreasing order of difficulty. Like first, we do like we build our maps manually with GIS data, or we could use like a R source code for Scanclim, and finally a version of this code that is embedded in an R4EU Scanclim, R4EU platform, which is also like is the same as Scanclim. So it's very important to notice that we have these 3 different methods. In order to help our external contractors, for instance, which may not be familiar with GIS data, to be able to actually proceed and produce these maps. And the final outcome is presented here. So this is a screenshot taken from the Scanclim embedded in R4EU platform, which is online and already available also for you if you log in with the credentials. And this is how it looks like. So we have this full map with the distribution data represented here either as dots or as with highlighted the boundaries of the administrative units that are showing the presence of the pest, and then the climate that in which this pest was detected that also appears to be present in Europe. For this was all for what concerned the pest categorizations. Now I will leave the floor to Eugenio for the second part about the quantitative pest risk assessment.
Okay, so thank you, Alex. So as you can see from this slide, the methodology of the quantitative pest risk assessment follows partly and the one that Alex just presented you on the pest categorization, but it expands certain topics and certain parts of this analysis in order to have a more in-depth analysis of the climate suitability. So the literature search works completely the same as it was for the pest categorization, but it is in the data extraction, as you also saw before in the outline of our methodology. Here we increase the number of information that we retrieve, because still we make use of the distribution and the host range as it was for the pest categorization, but together with the working groups and the scientific officers that are working on the development of pest risk assessment, we designed a framework in the distiller platform that from one single search string and one single data extraction could be used to answer to all the different steps that are involved in the quantitative pest risk assessment. Therefore, we, together with our colleagues, record information on pest biology, on impact spread, and on the control methods added to the distribution and host range, which are already collected in the pest categorization. So on the right, you can see the first product of this data extraction, which is a Prisma diagram. And now I will go through each of these subtables, we could call them, that we create, to show you a bit more in depth the information that we retrieve. So starting with the distribution data, as for the pest categorization, we only collect natural occurrences that have been detected or observed. So I wanted also to point out that all of this is implemented in the distiller software, which makes things run smoothly and fast in the data extraction process. So moving to the host data, here we designed the questionnaire to collect both the scientific and the common names, plus we gave the possibility to the reviewer to insert possible uncertainty in the host record or additional notes. And moving to the live stage, here the peculiarity of this and the following forms is that we were able to relate the host name that we recorded in every paper to the data extracted in this and in the following subtables. Therefore, there was a one-to-one correlation with the information and the host for which this information was extracted. So for the live stage, we record the live stage of the pest that was observed on the host, and also the host organ in which this specific live stage was observed. In this way, we are able to understand a bit the behavior on the host of the pest that we are looking for. Following this, we move to the impact that, together with the spread that is in the following slide, is based on the EFSA priority pest project methodology. And here we are collecting all those information that are defined as tangible effects that the pest produces on the host. Therefore, the incidence, the losses, and the symptoms that are recorded. The same happens with the spread, where we record information of, for example, flight capacity, spores dispersion, wingspan, and movement of the pest, and still is based on the priority pest methodology. So finally, we also included the control methods, because it happens often that we perform analysis also on the management and control methods inside the quantitative pest risk assessment. And here we can provide a correlation between the host, the type of the control method that is used, and also the explanation of the practice used. So to wrap up, the deliverables of this of our quantitative pest risk assessment methodology for the climate suitability comprehend a climate suitability report in which we describe all the steps and the methods that we used for completing the literature search, the model that we used, and the analysis that we perform. Then we also create a series of data extraction files which contain all the information that were retrieved from the subtables that I just showed you, and each of these information is connected to the paper from which it was retrieved. So in the master file, we index and record with a unique identifier all the references from which we performed the extraction. And finally, we also create a geopackage that contains all those geospatial layers that were either used or produced during the analysis. So if you notice that I didn't spend much time on the analysis of the climate suitability, because these are extremely specific to every quantitative pest risk assessment that we produce. Therefore, I wanted to show you the an example of climate suitability analysis to help you understand better what type of analysis we use in our work. So I will present you the case of Elasmopalpus lignocellus, which is also very interesting because it includes also an analysis of the climate change scenario. So to meet the pest, Elasmopalpus lignocellus is a polyphagous moth that is reported to feed on more than 27 families, but the most important host from the economical point of view are the Poaceae family, the Fabaceae and Asparagus. It is a pest that that likes dry and warm temperatures and is in the EcoAlert list since 2019. So again, we performed a systematic literature search, in this case consulting the EcoGlobal database, CABI and SINAVIMO to retrieve all those scientific and common names that we used to build the search string, which then we launched in Scopus and Web of Science. So after duplicate check and our final number of references was analyzed with the methodology that I just explained you. So as you can see here, the first products are the distribution, that here you can see we divided in observations at the coordinates level and at on the left, and on the right at the administrative unit level, where observation at the point location were not recorded. And from this we built the Köppen-Geiger climate matching map. So at this point, we are following exactly the methodology of the pest categorization, but now we dig more into the analysis of this pest. And for this specific quantitative pest risk assessment, we used Climex to perform further analysis. So here you can see the parameterization that we used for Climex, which was done following both information retrieved from the literature and tuning the model to match the distribution of the pest. So added to that, we also, given the biology of the pest, included data on irrigation and included in Climex as an irrigated scenario. And the data from irrigation, we retrieved them from Meyer et al. And we upscaled the database, the maps produced by Meyer et al. To match the same resolution of Climex, and applied the irrigation scenario only on those pixels that are highlighted. This is only for Europe, but we applied it globally. Only on those pixels highlighted in the picture on the right. So here you can see the result of our Climex analysis, and as you can see, the Mediterranean basin and parts of inland Spain and Portugal have an ecoclimatic index which is considered as favorable for the establishment of the pest. So, as I said, we also included the climate change scenario in our analysis. Therefore, we investigated with the Climex model the potential effects of climate change in a relatively near-term period from 2040 to 2059, centered on 2050, for the RCP scenario 8.5. So to perform this analysis, we used 4 regional climate models, and the output of these 4 climate models were used as input for the climate simulation. In this way, we were able to create and to compare the outcome of the simulations done with the historical to the one made with the future climate data. And we recorded the increase in the area of potential establishment. So here you can see the results of our analysis. In yellow, you have is shown the area with an ecoclimatic index higher than 30 for the historical data simulations, while in red you see the one for the climate change scenario. And as you can see, there is a high increase, especially in Spain and in France. So added to that, we also included the degree days calculations that helped us to estimate the potential number of generations in Europe compared to the one observed in the areas of pest distribution, and we analyzed it both for the historical climate data and the future climate scenario. So with this, our analysis is completed, and now I wanted to update you a bit on some more practical terms of our methodology. So here, for example, you can see the time estimation for completing our data extraction, which is in the risk assessment one of the most time-consuming sections. And as you can see here, on average, to retrieve all the information that we presented today, per reference, it takes averagely 16 minutes. So here you have a projection of what happens according to the and how time increases according to different number of references to be analyzed. Furthermore, I also wanted to show you some numbers of our team. So as Andrea said, it's the 3 of us that are in this room, but we also have some contracts with external experts and a grant with the CREA Bologna that is starting soon. And here on the right, you can see some numbers of our team. So we delivered, starting from January 2023, 19 pest categorizations. We adopted 3 quantitative pest risk assessments. We have 2 quantitative pest risk assessments ongoing and 1 that will start very soon. And up to now, we processed more than 9,000 references and recorded 8,000 observations, with a number of 79 maps produced overall between pest categorization and PPLA. Here, you can see a bit the response, the interactions that we receive in Zenodo with our publications, which, according to us, are fairly positive, because up to May 2023, we collected 440 views and 260 downloads of our data. Therefore, we encourage ourselves, but also the scientists performing climate suitability analysis to share their data as we do. Then this would allow a nice exchange of and growth of the scientific community. So coming to the future development, we are designing a structured geospatial database for the management of all the data that we collect. And furthermore, we are evaluating the use of remote sensing in our analysis, especially towards the host physiological development and its distribution, together with the evaluation of the potential impact of extreme events on the pest establishment. And finally, we are creating what we call the seed project, which is a user-friendly set of tools that will support the climate suitability analysis from our side and also from the member states of the European Union. And with this, we thank you for your attention, and we open the floor for questions.
Let me say just give you some additional information before opening the floor to questions. So what Alex and Eugenio has shown is a summary of most of the activity that we develop to support the pest risk assessments for what that concerns the climate suitability analysis. What is important to understand is, which is related to the way we work, is that all the analysis that we do, all the tools that we are using, they are the decision on which tool and which analysis are done are taken in collaboration with the member of the working groups that are of EFSA that are developing the pest risk assessment, and of course also with our plant health panel. And so this is why one important activity is also related to the development of tools to support the activity of climate suitability, but also to encourage and to allow our experts and our contractors to support us actively and to do themselves some exploratory analysis at the beginning of the climate suitability analysis process. And so, for instance, the scan tool, which was mentioned by Alex and also by Eugenio, is a tool written in R that was developed up to a certain extent in a way to be used by people that have no skills in GIS or coding in R, to allow them, for instance, to develop the climate type matching that we do using the Copenhagen climate classification. And these allowed us to allow the contractors that are supporting our activities to develop themselves the maps that we are mapping. And that tool, for instance, is available as a source code, but also as a web tool in the R4U platform. In addition to ScanClim, we have an ongoing internal project, which was mentioned by Eugenio, which is the SEED project, which is aimed at the development of tools, which will be available through the web, of tools to support climate suitability analysis, which will allow in a user-friendly way to develop maps, for instance, for agrometeorological indicators, or to run the species distribution models and additional tool that can be used in either in preliminary analysis of the climate suitability or for developing the final output of our climate suitability. And these allow us to work even more with external contractor supporting our activities. And that's it. So I leave the floor back to you, Frank, for any question or comment on our presentation.
Yeah, well, great. Thank you so much. Thank you so much. That was really, really interesting. And I don't see any questions in the chat. That doesn't mean people don't have questions. I would say if you do, maybe raise your hand, or actually if you drop something in the chat, I'll call on you. But if you don't mind, I'm going to go ahead and ask a couple questions. First, if you will put together a few sentences, I want to put like an abstract. This recording, it'll get a little. We'll clean up the front end so it'll just be limited to to the presentation and anything after. But if you guys will send me something, I can use this to promote it. This will end up on the IPRRG YouTube channel. Is that okay? Is that fine with you guys to promote it that way? Yeah, cool.
No problem. Yeah.
I think this is great. I mean, you went through the whole cycle of what we expect to see in this work. Everything, you know, you had multiple climate, multiple GCMs, and you know, RCP 8.5. That makes sense as an emission scenario. You showed those. You showed them under different conditions. You had irrigation in there. I mean, I feel like you guys, it was pretty soup to nuts kind of process. That was really impressive. I'm really glad, Andrea, what you said there about the seed project, because that was my one question as I was looking at the scripts under. Is it R for EU? Is that the platform? Like,
yeah.
Anyway, that was going to be my question: is are you trying to set up something so that people outside of EFSA and anyone under contract to EFSA, are you trying to do like a publicly available version of this? Is that like the seed project would be open to everybody? Is that the goal?
Yes, so the seed project is aimed at developing tools that will be available, of course, inside EFSA, and so in EFSA this will be used will be connected directly to our database to store the preliminary analysis that we do, but also to store, let's say, the final output, which will be connected in a geospatial database directly through our scientific outputs. But they will be open, yes, also to outside EFSA, because we want to develop something to support to support also the member states or the academia with tools that will do some, I mean, kind of basic analysis, but using stuff like GIS and code that if you don't have skills, they become difficult. So this is what we are aiming to support people without those skills to do those analyses, which can be helpful for risk assessment.
That's excellent. I think there's, okay, I have a couple follow-ups. So one, would you be willing, or are you okay with any links that you have in your presentation? Or I might actually reach out to you after. I think IPRRG, these are the kind, you know, we have resource links. We try to link to other, you know, so that people can get access to these resources. And I think, so would you be okay if I have a link to at least your outputs that people can get? It'll just point them there if they want to go get those.
Yes,
and you're okay with that? All right.
As far as I remember, there were two links. One was to the R4EU platform, which is
okay, great.
An platform where you will find different products there, and there is also this tool which include the scankling, and also a tool which allow the user to develop degree day maps and also number of generation maps based on data only for Europe, which are daily. It's based on daily data only for Europe. The seed project, on the contrary, will allow people to do to do analysis at the global level using a global data set.
Okay.
And the other link is to there's another platform where we are distributing all our outputs data, GIS in GIS format, but also in a JPEG format.
Okay, that's great. I think either I will follow, someone from the executive committee, and maybe me, will follow up, and we'll figure out some way to get links on the IPRRG website, and just so we can, this kind of cross-pollination is great. I think really, really important. Rose, did you have a question?
No, I don't, but I think what you just said about having the cross-pollination is incredibly important. This is something that we will be working going forward. It is exactly the same thing for some of the other other sources of information from third parties that we believe that will enrich the membership that we have. So I'm very grateful that you guys will be okay to share this with Frank, and we'll be able to put this on the website hopefully shortly. We do have also the ISF list that we needed to put in as well, Frank.
Yeah, I'm actually making a note on that too right now, to add that in there. I wanted to ask, go ahead, Rose, if you have something else.
No, I just say that there is no further questions on the chat, but if you do have some, please go ahead, Frank.
Yeah, I guess I have a couple questions. One is process. You have a lot of output this year. So I assume are you expecting this is the kind of level of volume of work that you can do going forward? Like this, what you presented, the more than 9,000 references in 2023. Do you hope that you can do this every year? It looks like you have a pretty good team.
I mean, it's for what we are doing, we are a little team, and I'm afraid this is going to increase in the next year. So this is why we also rely on external contractors. For instance, we are supported a lot in the pest categorization phase, and also for the quantitative pest risk assessment. Sometimes for specific cases, we try to collaborate with external experts to support us in the climate suitability analysis. For instance, one of the examples that was shown was the physiologically based population dynamics models that was developed by an external expert that was collaborating in the working group of the fall coloring month. And so we work like this. We personally try to support directly our working group of experts, or we are collaborating with external contractors, or we involve external experts in our working groups. But yes, it's thank you for saying that. It's a huge work, and you know, since during the last year, I've been on the most boring side of the coordination. The guys here are doing most of the analysis, the funny and the tough stuff. So, but yes, it's a lot of work, and we have unfortunately we have also a lot of ideas for the future. So,
well, that's good, and you know, Andrea, somebody's got to do the boring side, you know, that's just sort of how it goes. So somebody ends up with the responsibility. Well, I really think it's a nice, yeah, I understand the workload, but I'm impressed, and I hope you are able to sustain it. So this is kind of related to that. I want to ask, like, when you decide what set or you know what pests you're going to do next, like is this coming from your group of experts, or is it being informed by what EFSA needs? Or I guess that's the part of the process I wonder if you could elaborate a little bit, is like how you decide which ones you're going to do. You know, what is it, things that fall in EFSA's purview, or are you going to go and try and do something that you're like, oh, this is really an unusual case that maybe is looking at I don't know, an agricultural sector or a region or something that we, you know, that kind of thing.
Thank you for asking this, because this allows me to specify a bit better because this is related to the way EFSA in general work. So we do not, well, in 99% of the cases, we do not decide by ourselves the pest on which we perform the pest categorization or the full pest risk assessment, but we receive mandates from the European Commission to do the pest categorization or the full pest risk assessment. And this is based either on the activity of another team, which is the plant health monitoring team on the horizon scanning, or may depend also on, I mean, sometimes the interceptions are triggering the request for pest categorization or full pest risk assessment. Or sometimes we are asked to update our scientific opinion in relation to specific cases which might be are considered quite relevant and potentially harmful to for the European Union.
I mean, I think it's, you know, overall what you're doing is great, and I think however you get there is great. Whatever one you want to look at, that we found that not every pest that anyone is looking at ends up being incredibly relevant right now. I mean, climate change is driving that need to look. I mean, it's sort of baked into the question. If you're doing climate suitability analyses, a changing climate kind of dictates we got to look at everything at least one more time, probably multiple times. So I don't think you'll have any shortage of needs from any from within EFSA or anywhere. I think it's going to be it's an endless task. So you know, I'm glad to see you're digging in, and I really appreciate what you've done. And I don't see any more questions, so I will now let you off the hook unless you have some other anything closing that you want to say. I will close from the IPRRG side, say thank you again so much for doing this. We really appreciate it.
No, thank you again, you for inviting us and giving us the possibility to the opportunity to present our activities, and also to give you some preliminary information on something that will be made public and available to external analysts too. So thank you very much. Really appreciate that.
Okay, well, we will, I guess we're about to close here. Rose, do you have anything that you want to say from your side? I will say we should go ahead.
I just would like to say thank you so much to our presenters, and it was really very interesting, and I'm much more curious than what I was before. There's a little bit outside of my expertise, but I think it was lovely. It's very good to be able to show this to all of us, what the work that you guys are doing. So thank you so much for that.
Thank you.
Thanks.
All right, well, with that, we will close. I will remind anyone on this call to keep an eye out for information about IPRRG's annual meeting in Nairobi. So IPRRG 2023 will be in Nairobi, Kenya. It is the last week of September. The dates are the 21st. I'm sorry, it might be the next to last week. It is September 21st of September through 24th of September in Nairobi. You can already submit an abstract. The abstract submission form is open on the IPRRG website. Again, go to pestrisk.org and you can visit there and look for the tab that says IPRRG 2023. There will be more promo material on that coming out very shortly, I hope. And other than that, again, thank you so much, and signing off for now, and keep also look out for more about the next webinar. Thanks again. Thanks, Rose. Thank you, gentlemen. That was very wonderful, and we hope to see you again, hopefully in person at some point.